Okay. So Robin Waterman is currently a PhD candidate at the Michigan State's Kellogg Biological Station in the Department of Plant Biology and the Ecology, Evolution, and Behavior program. Robin began her academic career at Wesleyan University in Connecticut, where she initially received her BA in Biology Environmental Studies, and that's where she began her research in the laboratory of Sonia Sultan. She began researching the transgenerational effects in plants, first studying the effect of parental light conditions on offspring fitness. And these findings were published in the Philosophical Transactions of the Royal Society B. Robin continued on in Dr. Sultan's lab for her master's degree after this, researching the impact of parental environment competitiveness on offspring growth in a smart weed. So following along the same lines. And Robin published these findings in Ecology. And then in 2020, Robin joined Dr. Jeffrey Connor's lab at Michigan State University, researching the genetic and phenotypic effects that occur in Canadian horseweed during successful invasions of agricultural fields. So very interesting. So much impact could come from this work for the farming industry. And Robin has published one of her chapters in SSE's evolution. Finally, Robin has contributed to DEI work, including a paper on how historically excluded groups are impacted by peer review bias. And this was published in Nature, Ecology, and Evolution. And so with all of that, I will turn it over to Robin for her talk, Investigations of Adaptation to Agriculture in Horseweed.
Thank you very much for that introduction, and thanks everybody for coming to listen. So with that, let's get started. I want to start by talking about my motivation for studying this topic. So I think it's pretty striking that about 16% of Earth's habitable land surface is devoted to crops. And so agroecosystems really are one of Earth's major habitat types. And within these habitats that we rely on so much for our food, weeds seriously reduce the yield of our major crops when they're not controlled. The issue is that current control methods are often unsustainable and expensive. So my hope is that a better understanding of how weeds evolve and plastically adjust their traits will help inform management strategies for the future. So before we talk too much about agricultural weeds, I want to start by defining them. So I'm defining a weed as a plant growing in a disturbed habitat where it's not wanted. And so an agricultural weed then is an unwanted plant that's growing in actively cultivated habitats. And for this talk, I'm focusing on weeds of row crop fields. Largely based on comparisons of weedy and non-weedy species of the same genus, there are several traits that are commonly attributed to agricultural weeds. These include rapidly developing to maturity, producing seeds that can remain dormant in the soil until favorable conditions arise, reproducing through multiple modes, including asexually, sexually with pollen from other plants, and sexually with self-pollen, and a high rate of that last mode. Releasing allelopathic chemicals that negatively affect neighboring plants, and being resistant to herbicides. However, with the exception of herbicide resistance, it's generally unclear how widespread or important many of these traits are, and how unique they are to agricultural weeds. And we might expect agricultural weeds to have evolved adaptations because crop fields can impose a variety of novel selection pressures, including supplemental water through irrigation systems, regular spacing of crop competitors and weeding between these rows, chemical inputs of fertilizers and pesticides, and the clearing of biomass at harvest. For this talk, I'm primarily focusing on this last factor and how it might impact the life cycle or phenology of plants. I chose as my study system a plant called Canadian horseweed, or Canyza canadensis. It's native to North America. It has a high selfing rate, and it produces these wind-dispersed, single-seeded fruits on each flower head. One of these is shown blown up in this picture. It's a problematic weed of many row crop systems, but especially in no-till. And it can be a winter or summer annual. And that's what we're going to talk a bit more about. So horseweed develops in what's known as different growth types. The first is the upright growth type, which is a summer annual. And what that means is that it emerges in spring, immediately grows upright, forming its flowering structures, and reproduces in summer or early fall. In contrast, what's known as the rosette growth type develops as a winter annual. So it emerges in the fall, forms a low-to-the-ground rosette to overwinter. Then it bolts upright in the spring and produces seeds in the summer. This induction of flowering only following a period of cold is referred to as vernalization. Other prior studies have observed an increase in upright types in the field, at least in Michigan. And the hypothesis is that the increase in the upright type is due to associating with summer annual crops. And this might make some sense because we know that in the US, the vast majority of crop acres are summer annual crops, namely soy and corn. However, the specific environmental cues are largely unknown. And so these alternate strategies and their potential to influence ag field adaptation really fascinated me, and I carried out a series of experiments to try to learn more. My three main research questions were: first, do horseweed plants from agricultural populations versus non-agricultural populations show trait differences indicative of a shift to the upright summer annual type? Second, I asked whether plants from agricultural populations are locally adapted to the summer annual crop environment, and whether any fitness differences I found could be explained by alternate growth types. Third, I investigated what environmental cues and signaling pathways could trigger different growth types. Starting with question one, so based on my hypothesis that ag plants have shifted towards the upright summer annual type, I predicted a series of quantitative trait differences. I predicted that they would show an accelerated life cycle, emerging faster, flowering faster without the need for a cold temperature vernalization period, growing taller at a faster rate, but attaining a smaller height by reproductive maturity given this shortened life cycle. In addition, I predicted that when looking at growth types categorically, I would find a greater proportion of upright types for agricultural plants compared with non-agricultural plants. To test this question, I carried out an experiment largely with the help of one of my REU, or Research Experience for Undergrad mentees, Brooke Catlett. So I collected seeds in 2021 from both agricultural and non-agricultural populations of horseweed. I grew them all in a common greenhouse environment. And then in the following year, conducted a reciprocal transplant where plants sourced from ag and non-ag populations were then grown in both ag and non-ag growth environments. In this generation, I also grew plants sourced both from the common greenhouse environment and directly from the field to test for possible parental effects in which the environment a parent experiences during its life can influence offspring development. And so I'm going to be showing you data both from the common garden generation and the field reciprocal transplant generation. If we just looked at differences in means of parent plants, then the differences we found would be some combination of genetic differentiation and plasticity, but we wouldn't be able to tease these two apart. Whereas when we grow these plants in the common greenhouse environment, we can attribute mean differences to genetic differentiation. And furthermore, in the reciprocal transplant, we can look for both genetic differentiation and plasticity, where the same population grown in a different environment expresses a different trait value. So let's get to some results. First, I found that agricultural seeds emerged faster. And what I'm showing you here on the left in the red dot is plants from agricultural populations, and in the blue, plants from non-agricultural populations. And these are estimated marginal means, so accounting for family effects in this repeated measures design. Next, I looked at the transition to flowering, and I found that plants from agricultural populations were less likely to require a vernalization period. And so in the experiment, plants that had not shown signs of transitioning to flowering by the end of the summer growing season in August were vernalized in a cold chamber for 2 months and then moved back to the greenhouse for growth. So as expected then, based on these two, agricultural plants flowered faster, and this difference was entirely accounted for by the faster emergence and reduced proportion undergoing vernalization, since adding these two factors to the model with flowering time removed any difference between ag and non-ag. Next, I looked at height, and I found that agricultural plants had slightly greater height measured after the same number of growing days, indicating a faster growth rate and a difference that increased over time. But the overall difference was not significant. There was also no significant difference in how tall plants grew by the time they flowered. However, the means here were in the predicted direction, with agricultural plants being slightly shorter by flowering. Next, I looked at the categorical assignment of growth types, and I found that about 15% more plants from agricultural populations were upright compared with non-ag populations. The difference in proportions was only significant in the greenhouse common garden in 2022, which had the largest sample size, but the same pattern was found across these 3 experiments. So if we return to these quantitative traits, we can see that in every case, the direction of differentiation between ag and non-ag plants is the same as that for upright versus rosette plants. And this is true in each case: flowering time, height growth rate, height at flowering. And furthermore, for all traits except emergence timing, when growth type is added as a covariate to the models with source environment as the predictor, then source environment no longer has a significant effect. And this suggests that differences between ag and non-ag plants do indeed indicate an evolved shift to more upright summer annual growth type. Moving on to my second question, I asked whether plants from agricultural populations are locally adapted to summer annual crop environments, and whether any fitness differences I found could be explained by alternate growth types. I predicted that plants sourced from ag environments would have higher fitness when growing in their local ag environment type compared with foreign plants sourced from non-agricultural environments, and the opposite pattern in the non-agricultural environment. I expected this pattern to then be mirrored for upright and rosette plants. What I found was that the agricultural and non-agricultural plants had similar survival across environments, at least up until the summer annual crop harvest. But the ag plants were potentially more likely to flower by this time, although the difference was not significant, likely due to very small sample sizes in the field. If I multiply these first 2 fitness components, I get the total proportion of plants that survived and flowered. And this shows a similar pattern as just proportion flowered, given that similarity in survival rates. So although at this point there's no evidence for local adaptation, I want to note that these data are only until the end of the summer annual crop season. So in the ag environment, all plants died at harvest. But in coming weeks, plants are going to be scored for survival through winter in the non-agricultural environment. And if non-ag plants survived and then flower more often, then there could be local adaptation. Additionally, if you recall from the experimental design, I looked for evidence of parental effects on these fitness measures. However, there is no consistent pattern for plants from field versus first-generation greenhouse seeds. So parental effects do not seem to be playing a major role in this species. I next looked at these fitness measures in plants classified as upright versus rosette. And here we found higher survival across environments by rosette plants. And this is possibly because the rosette structure is designed for maximum survival, even through those harsh conditions, in order to make it through the winter. But since no rosettes flowered, these plants had zero fitness when looking at survival and proportion of survivors flowering overall. And if we match up the ag versus non-ag fitness with upright versus rosette fitness, it does seem pretty likely that that greater tendency for ag plants to be upright likely explains their increased likelihood of flowering, so that similarity in the red and the white bars. It's unclear why the rosette survival advantage did not translate to the non-ag survival advantage. So this means there could be other traits distinguishing ag and non-ag plants not captured by growth type, or that the relatively small boost in rosette types for non-ag plants was not enough to be statistically detectable. My third question, I wanted to learn more about what environmental factors and signaling pathways might trigger different growth types. So prior studies, Skramsky et al. In 2021, tested the effects of temperature, photoperiod, shade, soil moisture, competition, and cold stratification, all in the growth chamber. And they found that only cold stratification impacted growth type, causing 100% of plants to be upright. And stratification is when seeds are exposed to wet, cold conditions, either naturally outside or in the lab. And it's often necessary to break dormancy and stimulate germination. Horseweed does not require stratification for germination, but this study found that when it did occur, plants grew as upright types. When it did not occur, plants grew as rosette types. And this makes some intuitive sense because upright types germinate in spring. Receiving simulated winter conditions during stratification, causing plants to be upright does align with that expectation. And so in my experiments, I predicted that cold stratification, but using a wider sample of seeds than in this study, would still result in all upright types. So as I mentioned, with stratification, Skramsky found 100% upright type versus 100% rosette without stratification. And they carried out their stratification in the growth chamber and grew plants in the growth chamber as well. In 2022, I stratified all seeds, both outside and in the growth chamber, with the goal of getting all upright types. But I was very surprised to find vastly different results. So there was not an unstratified treatment in this case, but in the stratified for 4 weeks case, I found that the majority grew as rosettes rather than all upright. In 2023 and 2024, I attempted to more purposely replicate the Skramsky results. In 2023, I actually had to end the stratification treatment early since seeds started germinating in week 1. And regardless of whether they received this short period of cold temperature, all plants emerged as upright. So vastly different results. In 2024, again, exposed plants to either stratification or no stratification, but grew them in the greenhouse after the stratification treatment. And this time, the results were a bit more similar to what Skramsky found, with a slightly higher tendency for uprights than rosettes when stratified. However, the sample sizes here are very small. Overall, the results from these 4 experiments seem to indicate some role for stratification in growth type determination, but it seems clear that there's more to the story. It's likely some other environmental cue experienced either during seed maturation on the parent plant or during development in the seed germination environment interacts with stratification and likely accounts for why these results are so different across years. So given these results, I wanted to look at a few other factors and whether they might interact. So first, I wanted to look at light level. Although light level itself was previously tested and found to have no effect on growth type, what we wanted to do was test for interactions between light and stratification, since we hypothesized that plants that don't germinate until spring probably receive more shading in addition to cold temperatures while they're sitting outside, often buried under layers of snow or soil. Second, prior experiments used only seeds produced by upright parents. So we hypothesized that parent growth type might impact offspring growth type through the transmission of some parental effect. That is a transgenerationally plastic response to environmental conditions experienced by parents. And we predicted some degree of matching between parent and offspring growth types. Third, we shifted our level of analysis to try to identify a plant hormone involved in growth type differentiation. We chose GA, or gibberellic acid, as our most likely candidate, since it's known to play a major role in stem elongation and life cycle transitions in other species. For example, in wild radish, the application of GA in experiments causes plants to grow more upright and flower earlier, resembling the weedy ecotype. So we predicted that the application of GA early in development would cause plants to develop as the upright type. To test these 3 predictions, we carried out 2 related experiments. In 2023, we sourced seeds from agricultural and non-agricultural populations. We grew them in the greenhouse for a generation and sourced seeds both from upright and rosette type parents. We then put them into 2 stratification treatments, either cold stratified or not stratified, and then crossed that with 3 gibberellic acid treatments, either 0 applications of GA and only water, 1 application of GA, or 2 applications of GA, and the experiment included a total of 1,296 seeds. And this year, we're 2 of my high school mentees from the Kalamazoo Area Math and Science Center, Georgia Edmonds and Ishwari Bhatt, are carrying out a follow-up experiment. And in this experiment, we added a collection site from the same exact location as that used in Skramsky et al., just in case there is something particular about that population driving those results that they found. We again included a cold stratified and unstratified treatment. We crossed that with 2 light treatments in the greenhouse, a shade and a full light, and weekly applications of either water as our control or gibberellic acid. So first, I'm going to start with what we found to not be important. So we found no impact of the parent growth type. And from the 2024 experiment, we found no impact of offspring light environment. And these factors had no main effects or interactions with stratification as we had predicted. However, if we look at gibberellic acid, we found that repeated gibberellic acid applications cause plants to grow taller, which is an indicator of this upright growth type. So this graph here is showing estimated plant height, estimated marginal means plant height, and measured at 2 time points, one about 1 month and about 3 months into development. And we can see that there's a large overall effect of GA treatment. There's also an interaction with time point and block, and this was due to GA having a larger magnitude effect at the second time point in one of the blocks compared with the other, but the relative order of the points remained the same. In 2024, when GA was applied weekly, we found similar results where there was a large main effect of GA, and this effect increased over time. Then, if we categorically assigned growth types based on their morphology, whether they formed a rosette or not, they supported these results. And here's just a example photos of what these look like, these plants. So the categorical assignment showed that for the 2 applications of GA in the 2023 experiment and weekly applications of GA in the 2024 experiment, nearly all plants were upright. Whereas when plants received only water, in the 2023 experiment, none of them were upright, and in the 2024 experiment, only 30% were upright. So to summarize from my 3 main questions, I first asked whether horseweed plants from agricultural populations versus non-agricultural populations showed trait differences indicative of a shift to the upright summer annual. And I did find support for this in that ag plants emerged faster, were more likely to flower without vernalization, resulting in quicker flowering and slightly lower height at flowering. They possibly grew taller at a faster rate and were somewhat more likely to be categorically assigned as upright. However, the differences were less than predicted. And one possibility for this is that there is gene flow between agricultural and non-agricultural populations. Next, I looked at whether plants from agricultural populations are locally adapted to the summer annual crop environments and whether fitness differences can be explained by alternate growth types. And so far, the result is maybe. So the ag plants and the upright plants had similar or slightly higher fitness than non-ag plants across the growth environments by the end of the summer annual season, but I'm awaiting data from the following season to determine whether there is evidence for local adaptation. Third, I looked at what environmental factors and signaling pathways trigger different growth types. What I found is that cold stratification sometimes increase the likelihood of upright type, but other factors are likely at play. I also found that the application of the hormone GA increased the likelihood of developing as an upright type. However, there's no evidence that parent plant growth type or light level affected offspring growth type. Future steps for this project include collecting flowering data from the greenhouse plants in the current GA experiment, repeating the field reciprocal transplant with a larger sample size and earlier start, and conducting genotyping by sequencing on plants from agricultural and non-agricultural populations to try to look at whether that question of whether gene flow might be accounting for that smaller difference than expected between agricultural and non-agricultural growth types. And I'd like to give a special thank you to the Society for the Study of Evolution and their generous support through the Lewontin Award. And all of the people and listed on here were enormously helpful in providing advising, data collection, technical support. And thank you to all of you for coming and listening to my talk.
All right, great. Thanks a lot, Robin, for your talk and for your research. We have plenty of time for questions if anyone in the audience would like to type or unmute themselves. See if I can bring the chat up here. So I can start. D like how from the ag populations, how far away is the non-ag population relative to the dispersal of the plant? Like, if you were I'm imagining this area, uh, like that's just surrounded by farmlands, super far on either side. Maybe those plants have had less mixture with the non-ag population than somewhere on the perimeter or something like that.
Yeah, that's a good question. So as as I mentioned in the beginning, these plants are wind dispersed and they have those kind of sails on the top. And so there's not too much known about exactly how far they can go, but the thought is that they can travel pretty far distances. And so I think there is a lot of opportunity for gene flow, even like from even if the you thought like a population was pretty secluded, I think that would not prevent seeds from agricultural populations from getting there. But I think the interesting part is like, do they establish and succeed
Uh,
once they're there?
yeah, as they compete with the ones that are already established.
Yeah.
And then is dispersal easier in the ag environment, I'm imagining? Just it's like more open. The non-ag, maybe it's harder for them to get into a forest or something.
I think that I think that's definitely possible. We also know that there's going to be an effect of plant height on how far these seeds can get dispersed. So uh, that's one of the things I'm interested in is whether there are these differences in uh, overall plant height for these plants, and there haven't been measurements so far in the field of rosette versus upright types, but that's another follow up that I'm interested in. But yes, measuring dispersal would also be really fascinating. It would be quite a challenge, I think, for these tiny
Yeah.
little seeds. But I know there's a lot of cool research being done on how to measure or estimate dispersal distances.
I was just thinking if you could do it on a genotypic level, like see more signatures of the ag in an area surrounded by a lot of ag,
Yeah,
or more similar or more few more non-ag in the ag if dispersing to the ag is easier than the amount of ag in the non-ag. Does that
right. I see.
make
Yeah.
sense?
So like uneven direction of gene flow.
Yeah.
Yeah,
Like it would be a
that would
proxy
be
for it,
interesting.
but yeah.
Yeah.
Very cool. Well, does anybody else in the audience have any questions via chat or uh, speaking? Uh, if not, give a few seconds, but if not, I will end the recording and we'll thank Robin for a great talk and for getting us started this spring semester for the Greg Seminar Series. So thanks a lot, and I will see everybody next week.